And it is a pleasure now to host Cornelia Schwayer from the FMI in Basel. And Cornelia is going to talk about other aspects of morphogenesis in organoid systems. The title of your talk is Multiscale Integration of Cellular Context and Chromatin States in Tissue Patterning. Thank you, Cornelia, for coming.
Thanks a lot. So it's a great pleasure for me to be here today and to tell you about how the gut gets patterned. And I had, thanks Edouard, you already had a lot of introduction into many concepts that I will also talk about, and specifically also how emergence can be arising through patterning processes. And so in general, this can be observed in many different biological systems in development or also regeneration, where for instance, if you look at the vertebrates, you have a very important stage in development where you have the segment formation that comes through phases. Thanks. And so another example would also be when you have avian skin morphogenesis, where cells start out from a homogeneous progenitor layer, and at a certain point you start having patterning formed. Or during regenerative processes, as you just heard from Edouard, from a single cell, you will start forming a sphere, a symmetrical sphere, and at a certain time, at a certain point in time, you will see the first cell fates being specified here, the cells in white. Now, what actually happens in these different processes? Now, if we zoom into a single cell and then looking at the different scales, as you just heard, it's important to also look at many different scales in biological processes. We can see that the single cell gives rise to, or is usually in context with neighboring cells, and therefore generates its own local neighborhood. And on a tissue scale, you can also zoom out. Importantly here, the tissue, the complexity that you can see in tissue scale is much higher than the complexity of each individual cell. So how does that work? How does this complexity emerge? And so here, a lot of important work showed from many different labs that you have also tissue scale feedback that can come either from the entire tissue, also from the cell neighborhood, based on different extrinsic signals such as direct biochemical cell-cell contacts or also morphogens. Important factor here is also that it might, or it will, as I'll show you later, also depend on the cell intrinsic state. So meaning, what is the already gene regulatory program or the transcriptional program that the cell is already running independent of all the other extrinsic signals. And so on top of the cell intrinsic state that we'll be also focusing later today, what are the different types of extrinsic signals that a cell can face? And here, from the three examples that I showed you in the beginning, for instance, it ranges from biochemical to metabolic to mechanical cues. And importantly, these different types of information can also, or are observed to cross many different length scales. And we already had interesting questions about length scales. And here, just as an example, the cell is in has usually direct neighbors if in a tissue. Then on a larger scale, it can also face morphogens, but also mechanical signals. And so this shows that different types of information are usually also presented at different length scales. So what does this mean for actually understanding the emergence of complexity? We really need to be able to monitor and eventually also manipulate these patterning processes across scales. And so a model organism that I'm using is actually the mouse intestine, and it was already beautifully everything explained by Edouard. Briefly, the main bottlenecks in the in vivo mammalian system is that it's very hard to really track the regenerative process in the gut. So in the last 2 decades, the organoids have been emerging as a great model system, because what we can do here is to plate a single stem cell from the intestine of a mouse, and we can then track the cell division, the cell patterning in space and time. And here you see that this will form a symmetrical cyst, and after like 5 days you will already have an establishment of structures that are resembling the in vivo counterpart, But also importantly, not only the structure, but also the cell types will also mimic the in vivo counterpart. So really showing that this is a great system to use to address questions, because now we can actually monitor and manipulate the single cell behavior at high temporal and spatial resolution. And so the main question that I tried to address during my postdoc was to first understand how the cell decides here to actually become a blue or delta positive cell. So this cell will be patterned at around 16 cell stage of the spherical organoid. And importantly, later this cell will start secreting different growth factors. And thereby it will set up the stem cell niche, as you can see here. And the villus cells are usually the ones that actually do the job of absorbing and digesting the signals from the gut. So you need to have the first cell to appear in order for the entire system to work. Importantly, I just want to say that at this stage, the cells look all the same, right? But from previous work in the lab, we also know that at this stage, where there's no cell fate pattern yet, there already is a substantial cell-to-cell heterogeneity of YAP1. So what is YAP1? It's actually a known mechanosensor in many different contexts has been described, where, for instance, it can sense either low cell density or also stretch of the nucleus and the cells, and many other differences like ECM stiffness. But importantly, what does YAP do upon increased mechanical cues is that it can translocate to the nucleus, bind to transcription factors, and then, for instance, elicit proliferative programs, and thereby can also tune the proliferative system or the regeneration response. And so coming back to this slide where we showed that YAP is present in the cell-to-cell variable case, this is a concept that seems to be emerging also in other systems, because if we, for instance, look at the zebrafish heart development, or specifically in myocardial wall patterning, we see that an heterogeneity in cortical contractility will also instruct the patterning of the tube, or also, as also Edouard presented in their work, that in a mammalian embryo, temporal variability in cell division contractility results in robust morphogenesis. As well as in the organoid system, we see that this is a precursor. So now the question is, in the organoid system, how is this actually regulated? So how does the tissue transit from a heterogeneous YAP variable state, which is only a transient state, to a stable cell fate patterning of delta? And so I just show you one staining just to understand. Here we played from single cell, we go to the 4 cell, 16 cell stage and more. And what we see that YAP initially is present in nucleus, so active in all the cells, and then some cells start to turn off YAP. And so this happens at around 16 cell stage. And interestingly is now, if we look at all the delta positive cells, we actually see that majority of the delta positive cells have high YAP activity. However, there is a big mismatch between the number of YAP active cells, as you can see here, compared to the delta positive cells. So what is the regulatory principle in this transition? And specifically, how does the gut or the tissue go from a metastable heterogeneity case to a stable cell fate patterning? To understand this, we collaborated with David Bruckner and Edouard on this project to really understand the regulatory principles. And in order now to actually get access to the information on multiple scales, as I tried to mention in the beginning, it's now important for us to get access to single nucleus, cell and tissue scale features. I generated a machine learning algorithm where we can now really segment all these different features in 3D and throughout the regenerative process from several thousands of cells. Now, with this high content data in hand, we can start actually looking at what happens during regeneration. And so we can now also get measurements of heterogeneity, such as coefficient of variation, for instance, being one of them, where we can start seeing that it's actually increased with the emergence of delta. And now, what can we do with this type of data was to feed this into a model. And here, I'm showing you, we use the organoid data. And here, every sphere represents a single cell with its connectivity or neighborhood connections. And we use this to actually feed into a lateral inhibition model. So briefly, what happens in the lateral inhibition on 1D is that through initially you don't have any patterning, but through small stochastical differences, you will see that some cell will manage to get delta positive and thereby can successfully repress the neighbor from obtaining the same cell fate. Now in 1D, this generates a typical salt and pepper patterning, but now if we go to the sphere, which is the intestinal organ system that we wanted to understand, you see already that the neighborhood also information changes quite drastically. And on top of inputting the heterogeneity data from organoids, we use perturbation experiments using YAP inhibitor and YAP activator to constrain the model. So what do we see here in inhibition or activation is that in both cases we see a decrease in heterogeneity, and interestingly, either up or down regulating YAP will result in a failure in patterning. And this I will come back to this point in a few slides. So using this perturbation data plus the heterogeneity data of organoids, a more simplified version of the model actually looked like this, where we need to have, like simplified two cells, a delta positive and a negative cell, where we see that YAP activity primes delta, and delta needs to undergo a self-activation mechanism, and it of course inhibits its neighbor. So now from biological data, we couldn't find really a direct connection between YAP and delta, any binding for instance. So therefore we needed to, in order to explain these ratios, we needed to integrate an intermediary factor that we call factor X in between YAP and delta. And on top, to actually understand what is this self-activating loop that delta is showing here, we needed to go through neighborhood information, because delta itself was unlikely to really upregulate on a single cell level. But here we see that delta activates notch and thereby represses factor X in the neighboring cell. By this double inhibition, you will get a self-activation. And so what is this model? What is an initial output, for instance? It could look like this, where we see cells as spheres, and we actually can observe the different patterning mechanisms, where we can validate this with the data as well. So the first question we asked was, what are the stable states that the cells can attain during regeneration? And here we got inspired by the Waddington landscape, where you, well, I think this will also, and has been mentioned already, where we have a ball rolling down a landscape or a marble into different basins, and these basins represent the different cell fates. And so here external signals actually shape the landscape. Now for our system, for the intestinal regenerative system, the question was, which are the extrinsic signals, but also how does the intrinsic cellular state, in our case the YAP state, form or inform the cell fate decision-making process. And so this I show you here, the 3D manifold. On the y-axis is the specification of delta. On the x-axis is actually the lateral signal. And what I mean with the lateral signal is really the inhibition, the inhibitory inhibition from the neighboring, or the neighboring cells that the cell experiences. And on the z-axis is the intrinsic YAP state. So here we now have the separation of external signals as well as the intrinsic cell state. And we can start asking, for instance, what happens, or how does a 16-cell stage organoid look like now on this manifold? Now, for simplicity, I will show you two cut-through sections to the YAP high and YAP low state. And so what we found is that cells are actually present at two stable states, so the delta negative and the delta positive state. Now, two things actually need to happen for the cell to transit from the delta negative to the delta positive state. So one being that cells actually need to be present in a low lateral signal, so where they receive a low repressive signal from the neighbors. And a second factor is they need to cross this dotted line here, which is indicating an activation barrier. So how can cells do this jump? Here I show you that by increasing their YAP activity, they can actually transit into an intermediate state. And from this intermediate state, they can transit to a delta positive state. So this is curious because on more abstract terms, this means that YAP high cells, they show actually increased sensitivity to the environment, while YAP low cells don't show this, are not sensitive to the signal, because they will always be delta negative. Another interesting observation, as it comes with bistability, as also has been already discussed by Edouard, that once the cells reach the second state, they will be stably maintained there. So this is what the model showed us, and this also implies that there might be a memory of a previous cell state. And to actually test whether this is true, that cells actually retain their cell fate in the system, we used, again, now a live imaging where we can follow the organoids over several days and can extract features on single cells and the neighborhood. But importantly for this question, we used a delta reporter line. And so here, if you look at the blue cells, these are the delta positive cells, zooming into this lineage tree, for instance here, every bifurcation shows a cell division. And then once actually a cell is specifying a delta positive cell here turns blue, they will always stay delta positive. So really showing that indeed it's actually true what we also found from the prediction from the model. Interestingly, another point that I wanted to make, where actually the model was super helpful to also explain the experimental data a bit better, was that in the case of wild type, you have heterogeneity, as I mentioned, right? Some cells are YAP high, they will likely be more likely to specify delta depending on the neighborhood, and some cells are low in YAP. Now, in both inhibition and activation, we see that we have a failure of patterning, which was quite puzzling for us, that at YAP activator, we also don't get patterning. But now looking at, for instance, what happens in this landscape, so we see here at YAP low, it's intuitive, okay, cells never manage to reach a certain level of delta, and that's why they never manage to jump this barrier, because they lack YAP. But on the other hand, in the YAP activator case, we see now that all the cells manage to go to this intermediate state. So they manage to express a certain amount of delta, but now all the cells compete with each other and mutually inhibit each other from actually transiting and reaching this delta positive state. And so on the other hand, I showed you now how the particularities of the model, but now what is this factor X? Because for now it was mostly descriptive, but now we can see and inform what is the missing link here. And so we thought, or we hypothesized, YAP is, as I showed you in the early slides, it can interact with transcription factors, and also is known to change on the chromatin state of a cell, for instance. So now we thought it could be a transcription factor. Now, how can you identify transcription factors? So here we used an unbiased approach, where initially we performed single-cell attack sequencing. So here you actually check what are the chromatin regions in the cell that are becoming accessible or lose accessibility. And so I show you here on the left the UMAP on the different time points now in organoid regeneration, going down here and to this island. And the cell fate markers that actually show here, the gray cluster represent the unpatterned cells initially, and the blue cluster here represent the delta positive cells. Now, then we asked, what are the transcription factor motifs that become more or less accessible? And so specifically by identifying which transcription factor motif becomes accessible, we can then approximate maybe what could be the transcription factor that could actually bind these motifs. And so with this analysis, we identified that the cascade of FOXA transcription factors actually already became accessible here earlier than the canonical secretory regions. And so this really suggested to us they could be involved in this integration. Now, FOXAs, I show you here 3 different FOXA factors, and I start with the first one, where we thought, okay, let's test all 3 of them, and the FOXA1 was the one where we also saw that it actually precedes delta expression, so it's there already at 8-cell stage, and once delta comes up, it's also well localized. On the other transcription factors, for instance, FOXA2 and FOXA3, we found that there's much less enrichment, or it's much less present in the delta positive cell. We also had perturbation experiments, but I'm happy to talk about this later. So after all, I show you the interesting data on the CRISPR-Cas9 mutant line of organoids, where we found that if we get rid of FOXA1 transcription factor, we lack completely the delta patterning, which was really cool, because now this really allowed us to put FOXA1 here instead of the factor X, and have follow-up experiments to really understand whether this is really the factor that we are missing. And so on the other hand, we wanted to know whether maybe FOXA1 is not only necessary but also sufficient to trigger delta expression. And so we generated a FOXA1 overexpression line. And here we see a higher fraction of FOXA as expected, and interestingly also more delta positive cells. So really showing that FOXA1 is not only required but also sufficient. And here we have several other arrows, of course, that point towards or away from FOXA1. And I will mainly now show 2 more arrows, but happy to talk about the other ones in the Q&A session. So another connection or link that we wanted to test was indeed whether YAP1 is somehow priming FOXA1. And so from chromatin data of ChIP-seq, we also know that YAP perturbation or high YAP activity allows more binding of FOXA1 to the transcription, to the landscape, as well as on the antibody staining level, where we see look at the abundance of FOXA1. We find that in the case of YAP inhibition, there is a reduced FOXA1 levels, while in the case of YAP activation, we see more FOXA1 levels. And this shows, together with the chromatin data, that YAP1 indeed primes FOXA1 for later delta activation. And the final arrow that I want to now test and show you the results was to see whether indeed there is, if we break off the lateral signal, to see whether this inhibitory loop from notch is actually true on FOXA1. And so to hear what would be the expectation if we now take away this inhibitory feedback, we would expect more cells that are FOXA1 positive, right? And so this is what the model simulated looks like, where we see more of these spheres or cells will be showing high FOXA1 signal. Now, using in the organoid system, we used DAPT, which is a gamma secretase inhibitor. But long story short, what happens is that we can get rid of this lateral signal or reduce it. And in such case, we indeed see that experimentally we can manage to get more of these FOXA1 cells. So really also showing that this error is true, where FOXA1 is actually inhibited through the lateral inhibition. And so taken together, I showed in the last few slides that FOXA1 is indeed the factor X that we were looking for. And on one hand, it not only integrates the intrinsic cellular state, or for us, the YAP state, but it can also integrate the neighborhood information by dealing with the notch binding. And so this was all done in the in vitro system. So we use here the in vitro system to actually form a hypothesis, to really use unbiased ways to identify new candidates that we can then use and go back to the in vivo system, where we can specifically targeted test this identified transcription factor, for instance. So we asked, is this heterogeneity integration through FOXA1 also conserved during in vivo regeneration? And also here, we'll first show you what the model actually predicted or suggested us also to look for, where here is a 2D lattice now, and as also beautifully explained by Edouard before, we see here that cells usually take this hexagonal shape, and so these are mimicking cells, and we now input the information that we get from the organoids. Again, initially, YAP signal or activity is high, and then goes down or goes lower towards homeostasis. What will be the result here? On one hand, FOXA1 starts to get patterned once YAP already is sufficiently downregulated, but still heterogeneous. And we also found that there should be a peak of FOXA1 prior to reaching actually the homeostatic and also stable state, because here delta patterning will be maintained even though YAP activity is already downregulated. So we then tested this by using a mouse irradiation system. So this is used as a typical regenerative system. What happens here is that we can ablate highly proliferating cells within the crypt regions and then read out what happens 24-48 hours post-irradiation. And so in that case, as expected, we see that YAP levels are initially high and then go down towards homeostasis. Indeed, we also see this peak of FOXA1 at the 48 hours. And importantly, at the time point where the tissue reaches homeostasis, the patterning of the secretory progenitors is maintained, even though YAP is already downregulated. Also in line with the bistability that I mentioned prior in the organoid system. And so, okay, this is more like what happens during regeneration, like in a wild-type case. But now, what would happen if we would also affect YAP activity in the in vivo case? And so for that, again, from the model, what was the suggested phenotype was that if we increase YAP activity continuously, so through 24 and 48 hours post-irradiation, we lose completely the patterning of FOXA1, but also of the secretory progenitor. And so now looking at the in vivo system, where again we use a mouse irradiation system and use a LUT inhibitor, so in a way to actually and keep YAP activity high in the animal model, we indeed found that in such case there is a dramatic decrease in the FOXA1 positive as well as delta and secretory progenitors. So showing that indeed it's critical to have a certain level and heterogeneity of YAP also in the in vivo regenerative system. Now, in the last few slides, I showed you, okay, we have the model that was constrained by high content imaging data and perturbation data, and we could successfully predict the in vivo regeneration. Now, what happens once the tissue actually exits this regenerative response? So once the tissue reaches homeostasis, is it still possible that YAP can control or regulate the patterning at this homeostatic state? And so here, from the model, the prediction was, in case of homeostasis, you of course have patterning of FOXA1 and delta, but upon YAP activation, you would completely lose again the patterning effect, even though you have been already patterned before. So in that case, we also found that with the in vivo system, again with the mouse, we apply YAP activate at homeostasis, and indeed find that in such case we completely lose both the patterning on the FOXA1 and secretory markers. So showing that YAP still remains a control parameter also during patterning and for homeostasis. And this could also be interesting for cases of, for instance, cancer or disease phenotypes, where you also see that usually comes across with, or along with increased proliferation and mispatterning. Now, I showed you in these slides that in order to go from kind of a metastable cell-to-cell heterogeneous state to a tissue patterned unstable patterning case, how this is regulated, namely through a feedback where FOXA1 integrates YAP activity or the intrinsic cell state with also the neighborhood information. Now, I don't only want to discuss with you how this process happens, like on a mechanistic level, but also when does it happen. So what sets the timer of this, when the patterning is actually initiated? Why is this important? Because if you imagine if a tissue starts to get patterned earlier or later, it might really will have an effect on the pattern per se if there are no correcting mechanisms in place. So for instance, we know that in the intestine, they start proliferating in the beginning, and then at a certain space or time point, you actually have the patterning emerging. So the question was, what is this trigger? And so to address this question, we went back to the multiscale analysis pipeline where we extracted many different features. And let me show you some of them, which on one hand is the tissue scale features such as tissue density. And here we see that tissue density increases with regeneration. And interestingly, cells actually reach I call this the so-called intermediate tissue density, which is exactly when the first delta positive cell is getting specified. Now, importantly, this is also marking the moment where cells actually are present in the most heterogeneous state here, as shown as a peak in coefficient of variation of YAP compared to normalized tissue density. So what this implies, or what we wanted to ask next, is there somewhat of an optimal tissue density that is important for this heterogeneity establishment? And so to test this question, that was also introduced by Edouard, we used the bioengineering approach, where now we can, at the fixed circumference, we can actually put different amounts of cells to generate low tissue density or high tissue density states. And here you can also see an example of the nuclear stain. So using this system, we then functionally perturbed tissue density, and interestingly could find that again at this intermediate tissue density is where we have the peak of coefficient of variation. Now this looked very promising for the in vitro system, so then we wanted to know, is this also true for the in vivo system? And there, going back to the irradiation case and performing single nuclear segmentation, we also identified that the peak of coefficient of variation actually happens again at this intermediate tissue density. And this shows that in all the three systems, in vitro and in vivo, tissue density seems to act as a regenerative timer. So the tissue is proliferating, and once a specific or critical tissue density is reached, this will lead to heterogeneity establishment. Okay, but now this is all a tissue scale feature, right? Coefficient of variation. So what if you are a single cell here, what tells this single cell to turn on YAP versus the neighboring to not have YAP turned on? So on a single cell, how do you get to this heterogeneity? And so here, going back to the single cell data from the imaging time course, one feature that was striking again that showed very similar behavior in many different volume axis ratio shape features was that all of them became more heterogeneous, so coefficient of variation was increased. So suggesting that with regenerative time, a lot of cell shape features actually become more heterogeneous. And using now four central parameters, which was local cell density, axis ratio, size, and cell stage, we then generated a statistical model where we could also explain up to 40% of the observed YAP variance. So really showing that these four features already explain quite a lot of the YAP signal that we see. Now, we collapsed all these four features on a single axis to get a more intuitive way to see what happens between these features and YAP. And so, importantly here, we call this the morphoaxis, where we see that cells go from a highly stretched, big nucleus, low local cell density, to a high-dense tissue and low YAP activity. And interestingly, this really forms a sigmoidal shape, where we see that, again, at this very steep region, we see that this is exactly the state where the tissue enters this intermediate density. And this really suggests that small changes along this morphoaxis can already lead to big changes in YAP activity. And that also could explain why you have this heterogeneity establishment. Now, I told you today two things, or I wanted to address two questions. First, when is the tissue getting patterned? And this really depends on once the tissue reaches an optimal tissue density, and how is through this regulatory motif, where FOXA is the central integrator of intrinsic and extrinsic features. Now, using high content data, we managed to generate a cell-fate decision landscape that integrates these different aspects and could provide how mechanical cues are integrated with classical delta notch signal to pattern the gut. And so, as two last take-home messages, I wanted to also mention that, or coming back to the earlier slides where I told you that this might be a general principle where heterogeneity precedes a patterning process. In order to go closer to that question, we also tested whether this could be true in other systems or other species. So first, I want to show you the human small intestinal organoid data, where we established also the intestinal system from the organoid in the lab. And here, after plating single cells and looking at the regenerative time course, indeed we see that YAP is heterogeneous, and the first secretory emergence happens also around 20 cell stage in the human system. And interestingly, in the progenitor cells, they show also higher YAP activity compared to the negative cells, showing that there's really correlation and similarities between mouse and human system. And on the other hand, we were curious whether this is also true in other epithelial tissues. So then we went kind of down in the digestive tract, where we looked at regeneration in the colon. And again, in organoid-based system, where we see that indeed in colon after or during regeneration, there's YAP heterogeneity established, and in the delta progenitors, we see higher YAP activity compared to the negative ones. And these are all promising experiments, where again, we look at different species, but also within the digestive tract. And then as a last experiment that I want to share with you is we were wondering whether this is also true for other epithelial tissues outside of the digestive tract. And here we use mouse salivary gland organoids, again setting up a regenerative response, seeing that at some point when delta appears, we have YAP heterogeneity, where again delta positive cells show higher YAP. This is also in line with the work from Sylvia Frey's lab. So taken together, first take-home message really seems that this could be a generalizer principle where heterogeneity can drive patterning in different systems. And on a single cell level, I want to leave you with that, that it's, of course, the type of extrinsic information or extrinsic signals that the cell experiences is very crucial, but also the length scale they come with but importantly also the intrinsic cellular state. I showed you today that cells are not able to properly respond if they have a YAP low state, but they actually manage to respond once they have a certain amount of YAP activity. And so with this, I would like to thank the entire lab, specifically Sylvia, Edouard, David, and Priska for working together on this great project, funding sources, and I also want to announce that at the end of the year, we'll open my lab in Berlin at the Max-Dalbruck Center, where we will address how a complete regenerative behavior is regulated by studying the liver. And with this, thanks a lot.